Yesterday, last class, yesterday already discussing about the first part of the excretory system and their elimination. Yesterday up to the first you are starting to the yesterday urine formation. In the urine formation, how many steps are there? In the urine formation, the main process involved glomerular filtration. What is the first one? Glomerular filtration. The second one is known as a reabsorption. The third one is known as a secretion. Three different steps are involved to the urine formation. What is the first one? Glomerular filtration. This glomerular filtration takes place between the Bowman's capsule and lumen of the Bowman's capsule and glomeruli. It is nothing but afferent arteriole. It is nothing but afferent arteriole. It is indicated glomeruli. It is indicated Bowman's capsule. Between the glomeruli and Bowman's capsule, this process is known as a ultrafiltration. This process is known as a ultrafiltration. This ultrafiltration, there is nothing but glomerular filtration takes place between the glomerular blood capillary and lumen of the Bowman's capsule of the Bowman's capsule. Coming to the second step, reabsorption. The reabsorption takes place between the blood capillaries and renal tubules. See now, it is indicate peritubular capillary network. Peritubular capillary network. Now the reabsorption process takes place between the renal tubules to blood capillaries. Renal tubule to blood capillaries. That is the second step involved to the reabsorption. The useful materials, sodium, glucose, amino acids, these amino acids, potassium, different materials reabsorb into the vitamins into the renal tubules to the blood capillaries. Renal tubule to the blood capillaries. Now, what is the third step? It is involved to the secretion. The secretion means it is involved between the blood capillaries and renal tubules. Blood capillaries and renal tubules. Blood capillaries and renal tubules. See now, this step involved to the blood capillaries and renal tubules. Now, how many steps are involved to the urine formation? The first one, glomerular filtration, or also known as the ultrafiltration. It is located between the glomeruli. This glomerular blood capillaries between the lumen of the Bowman's capsule. That is the ultrafiltration step. It is located between the glomeruli and it takes place between the glomeruli and the lumen of the Bowman's capsule. What is the second step? Reabsorption. Reabsorption means this step is involved to this step takes place between the renal tubules to the peritubular capillary network. Come to the next one, secretion. The step takes place between the peritubular capillary network to the blood capillary to the that is known as the renal tubules. These steps are involved. The first step, urine formation takes place in the blood, which is carried out by the glomerulus. What is it? Glomerular filtration. See now, the blood capillaries originate from the heart. That is nothing but afferent arteriole. The afferent arterioles are modified into the glomeruli. The glomeruli gives rise to the efferent arterioles. What is it? Efferent arterioles. On average, 1100 to 1200 ml of blood is filtered into the 1/5 part. Now, what is the meaning of 1/5 part? 25% of the blood. 25% of the blood reaches into the glomeruli. The heart releases into the per minute 5 liters of blood. Out of 5 liters, 25% of blood reaches into the glomeruli. And the 25% in the 5 liters. How much? 1100 to 1200 ml of blood is filtered into the per minute. Filtered into the per minute. Each ventricle, that is nothing but stroke value, that is stroke value cardiac output 70 ml. The glomerular capillary blood present is caused by the filtration of blood through the 3 layers. That is 3 layers. The glomerular blood capillaries, it is takes place in the 3 layers. What is the 3 layers involved to the? What are the 3 layers involved to the endothelium of the glomeruli? What is endothelium of the this endothelium? Endothelium of the glomeruli. Glomeruli endothelium. What is endothelium? How many layers are present in the capillaries? The how many layers are present in the capillaries? Only single layer present in the capillaries. Arteries located. Arteries consist of 3 layers. Tunica externa, tunica media, tunica interna. Veins are consist of tunica externa, tunica media, tunica interna. But capillaries consist of single layer. Tunica externa layer is absent. Tunica media layer is absent. Tunica internal layer is only present. This tunica internal layer are also known as endothelium. What is it? Also known as endothelium and the epithelium of the Bowman's capsule. What is known? The epithelium of the Bowman's capsule. What is known? Epithelium of the Bowman's capsule. Epithelium of the Bowman's capsule. And also basement membrane between the 2 layers. It is the one first layer endothelium. Here epithelium. This layer it is indicate basement membrane between the 2 layers. Basement membrane between the 2 layers. The basement membrane between the 2 layers. It is the basement membrane between the 2 layers. These 3 layers, whenever the blood filtrated into the plasma only filtrated. The plasma only filtrated into the first one endothelium. Endothelium to the basement membrane. Basement membrane to the epithelium cells. Outer membrane cells. That is nothing but this basement first layer endothelium of the blood capillaries and epithelium of the Bowman's capsule. After then the epithelium of the Bowman's capsule they reaches into the filtrated materials reaches into the lumen of the Bowman's capsule. Lumen of the Bowman's capsule. Next one. The epithelium cells of the Bowman's capsule is called as a podocytes. Epithelium cells of the Bowman's capsule is known as a podocytes. Epithelium cells. These are the cells like this. Epithelium cells. Epithelium cells. The podocytes. See how this arrangement of the podocytes are located. Epithelium cells of the podocytes. What is podocytes? These cells look like a feet shape. The podocytes. The podocytes. The podocytes located in the epithelium of the Bowman's capsule, arranged in the intrinsic manner. The small pore gap located in the one side by side, they will arranged between the gap is known as the minute pores. That is nothing but slit pores. The small slit pores are located. The small slit pore they reaches into the filtration. The blood is filtered so finally through membrane. The almost on plasma except what is proteins. The large protein they cannot pass into the lumen of the Bowman's capsule. Lumen of the Bowman's capsule. And therefore it is considered called as a process called as a ultra filtration. The final end product of the lumen of the Bowman's capsule is known as the end product of the ultra filtration. What is ultra filtration? The amount of filtered kidney per the minute. How much? GFR, it is the approximately 125 ml per minute. How much per day? 180 liters per day. How much? 1 liters per day. The kidney of the have different regulatory mechanism. That is nothing but one such efficient mechanism that is known as a carried out the juxtaglomeruli apparatus. Now, what is the meaning of the juxtaglomeruli apparatus? Now, what is the meaning of the juxtaglomeruli apparatus? Juxtaglomeruli apparatus means it is indicate Bowman's capsule. It is indicate Bowman's capsule. The Bowman's capsule, it is consists of the what is afferent arterioles and glomeruli. What is this one? Afferent arterioles. This one afferent arterioles. This one indicate glomerular blood vessels. What is glomerular blood vessels? See here one such efficient mechanism can juxtaglomeruli apparatus. Now, what is the meaning of juxtaglomeruli apparatus? Here indicate three different types of cells. Three different types of the cells. What is the first cell? Afferent arteriole, outer surface. The cellular modification of the cells is known as the JG cells. What is the cells? JG cells. What is the cells? Juxtaglomeruli cells. Juxtaglomeruli cells. Juxtaglomeruli cells. Coming to the next one, if you observe the DCT, what is it? DCT. The DCT consists of the transverse section of the DCT. This DCT consists of the macula densa cells. What is the cells? Macula densa cells. Macula densa cells. It is the second one. The first one, juxtaglomeruli plus this macula densa cells. Juxtaglomeruli plus macula densa cells is known as the juxtaglomeruli apparatus. What is the indication of the juxtaglomeruli apparatus? Juxtaglomeruli JG cells located in the efferent arteriole. Efferent arteriole. Why they indicated the JG cells, juxtaglomeruli cells? It is located outer surface of the afferent arteriole nearer to the glomeruli. Nearer to the glomeruli, they are located in and also macula densa cells located in the DCT. These both cells are indicated juxtaglomeruli apparatus. Juxtaglomeruli apparatus. This JG is a special sensitive reason for the cellular modification distal converted tubules. The distal converted tubule consists of macula densa cells. JG cells consists of the juxtaglomeruli consists of the JG cells. Afferent arteriole outer surfaces consist of the JG cells. This afferent arteriole location of the dead contact. See, whenever DCT contact to the DCT contact to the DCT contact to the DCT contact to the afferent arterioles. But according to in our textbook, they are not indication. According to in our paragraph, they are indicated the DCT contact to the afferent arteriole. Whenever the blood pressure is decreases, these macula densa cells are activated. Whenever seen here, the fall GFR. Fall GFR means whenever the blood pressure is decreases, GFR decreases. That is nothing but glomerular filtration rate decreases, BP also decreases. Whenever the GFR rate is decreases, glomerular blood pressure is decreases, and the blood pressure also decreases. At the time, the macula densa cells are activated. What is it? Macula densa cells are activated. Whenever the macula densa cells are activated, this DCC contact to the afferent arteriole, this macula densa cell activation of the JG cells. What is it? The macula densa cells are activation of the JG cells. Whenever the JG cells are activated, the JG cells produce one important, that is known as the renin. What is it? That is produced to the renin. Whenever this renin is it is produced into the blood, it is more released into the blood. In the blood, angiotensinogen. What is it? Angiotensinogen. Angiotensinogen. The angiotensinogen, it is activated into the angiotensin 1. Angiotensin 1 activated into the angiotensin 2. This angiotensin 2 act on the adrenal cortex as well as ADH. Whenever they act on the ADH, reabsorption of the more water molecule. more water molecule. Whenever the reabsorption of more water molecule, sodium ions from the renal tubule to the blood capillaries, the blood pressure is normalized. Blood pressure is normalized. Cells similar to the renin. Okay, what is renin? That renin, which is similar to glomerular blood flow into the GFR back to the normalized. Back to the normalized. See now, a comparison of the volume of the filtered 180 liters per day, 1.5 liters are the urine. So this means remaining 99% of reabsorption into the is called as a reabsorption. Now only reabsorption into the 99% reabsorption. Only eliminate to the outside the body 1.5 liters of the per day. 1.5 liters of urine eliminated into the body. Means out of 180 liters, only 1 liter only eliminate into the outside the body in the form of urine. Remaining 99% of the urine is filtrated into the renal tubule to the blood capillaries. Renal tubule to the blood capillaries. Reabsorption process takes place between the, it is occurs between the renal tubules to the blood capillaries. Renal tubule to the blood capillaries. Renal tubule to the blood capillaries. The renal tubule to the blood capillaries. Renal tubule to the blood capillaries. The useful materials reaches into the renal tubule to the blood capillaries. The tubular epithelium cells are different segments perform the either active transport or passive transport. Active transport, they utilize the ATP energy. Passive means the directly they are not utilize any energy molecules. For example, such like glucose, amino acids, sodium in the filtered reabsorbed active through the nitrogen based absorbed into the passive transport. Synonym: glucose, amino acids, sodiums. These are the reabsorbed into the active transport. They are asking, okay, sodium, amino acids, glucose reabsorbed into the, that is active transport. Remaining waste material absorbed into the passive transport. Reabsorption after water, it is almost occurs in the passively. Initial segment means PCT. Initial segment PCT, segment of the PCT, initial segment after the DCT, collecting that the loop of vinyl, it is absorbed into the active transport. During the urine formation, the tubular secrete into the like H plus K plus ammonia into the filtrated. The tubular secretion also important role for the, whenever the reabsorption takes place between the renal tubule to blood capillaries, at the same time the secretion also takes place. The secretion also takes place between the blood capillaries, that is the peritubular capillary network to the capillary network to the BCT, network to the DCT, and also collecting that to the, they enter into the blood capillaries to the peritubular blood capillaries to the renal tubules. At the same time, reabsorption as well as secretion, it is helpful for the maintain the balance of the, maintain the ionic and acid balance. Ionic balance. See now, proximal convoluted tubules. And the proximal convoluted tubules, they are consist of the proximal convoluted, what is the PCT is lined by the simple cuboidal epithelium surface. And the PCT, it is lined by the simple, what is the simple cuboidal, what is the simple cuboidal epithelium, what is the simple cuboidal epithelium. This simple cuboidal epithelium, see, you can observe the simple cuboidal epithelium. It is simple cuboidal epithelium like this. Simple PCT, you can take observation of the PCT. The PCT consists of the simple cuboidal epithelium. Outer surface is the brush border appearance. Nearly almost all utensils, 70 to 80%, 70 to 80% of electrolytes and also water. What is water also reabsorbed into the PCT. PCT, it is also maintain the pH and ionic balance of the selective secretion of the hydrogen ions. whenever the reabsorption as well as secretion. Reabsorption as well as secretion. What is the secretion? Hydrogen ions. Next one, ammonia ions. Next one indicate potassium ions. These are the secretory material to the blood capillaries to the renal tubules. Here the renal tubule to the blood cap, there is a reabsorption and also absorption to the bicarbonate ions also. Okay, coming to the next one, Henry's loop. Reabsorption is minimum ascending limb of the loop. This reason plays a significant role in the maintenance of the high osmolarity medullary interstitial fluid. The descending limb of the loop of Henry only permeable to the water. What is water? It is important. The descending limb of the is a water reabsorption, but almost all impermeable to the electrolytes. This concept filtered into the movement of the down. Ascending limb of the impermeable to the water. Ascending limb impermeable to the water, but the transport in all electrolytes actively or passively. Ascending limb. Therefore, the concept filtered upgrade into the dilute into the passage of electrolytes into the medullary fluids. Distal convoluted tubules. What is distal convoluted tubules? The distal convoluted tubules are synonym. The distal convoluted tubules are condition reabsorption based on the condition. For example, at the time of the all useful materials, all urine can continuously enter into the outside of the body. The condition is the dehydration process, vomiting process, loose motions time. This one, DCT, it is the reabsorption of the renal tubule to the sodium, water from the renal tubule to the blood capillaries. At the time of the condition, based on the conditions, and water takes from DCT, have the reabsorption of the bicarbonate, selective secretion of the hydrogen and potassium ions, ammonia to maintain the pH and balance. Whenever the reabsorption takes place, at the same time, secretion also takes place. Keep in your mind, one point remember in your mind. What is the first point? Reabsorption takes place between the renal tubule to the blood capillaries. Secretion, blood capillaries to the renal tubules. Coming to the diagrammatic representation, see now once. The diagrammatic representation of the scene once. First one, see PCT. What is this one? PCT. These arrows indicate. These arrows indicate reabsorption. This arrow indicate reabsorption. The reabsorption means renal tubules to the blood capillaries. The renal tubules to the blood capillaries. The fluid is CO3, bicarbonate, sodium chloride, water, nutrients, potassium reabsorbs into the renal tubules to the blood capillaries. These arrows indicate secretion. See now, these arrows indicate secretion. This arrow indicate secretion. The secretion means blood capillaries to the waste material. That is nothing but hydrogen and ammonia. They are secreted into the blood capillaries to the renal tubules. Blood capillaries to the renal tubules. You can indicate H plus and ammonia, creatinine. The creatinine drugs. This all waste material reaches into the blood capillaries to renal tubules. Reabsorption. This arrows. Reabsorption means see renal tubule to the blood capillaries. They reaches. This one indicate potassium, hydrogen, and also ammonia. What is ammonia salts indicated in blood capillaries to the they reaches into the renal tubules. And this one also indicated renal tubules to the blood capillaries are interstitial fluid. It is also indicated renal tubules to the blood capillaries. They reaches into the this arrows indicate upper arrows indicates this arrows indicated reabsorption. This arrows indicated and it is also reabsorption. It is also reabsorption. And down these arrows indicate secretion. That is the difference now. See, it is the descending limb of the thin segment, water reabsorption, but almost electrolytes are no reabsorption. See here, water 70-80% water, nutrients, everyone is reabsorption here now. Coming to the thin segment, thick segment means only the electrolytes, no kind of reabsorption takes place in the water here now. Okay, here also sodium chloride, water, bicarbonate. These are the reabsorption. These are the reabsorption. See now. Coming to the collecting duct. Now this is a collecting duct means please indicate long tube action from the cortex to the inner parts of the large amount of water could be reabsorbed from this region produced with concentrated urine. Okay, now allow the pass a small amount of urea into to keep into the osmolarity to maintain the concentrated gradient. That's why to play the important role for the pH and ionic balance of the blood, selectively secretion into the selective secretion. See now, urea it is entered into the collecting duct to the interstitial fluid or interstitial space. They can reach us into the loop of NA. They maintain the osmolarity and also they maintain the osmolarity. Okay, they maintain the osmolarity. See now, the maximum water reabsorption takes place between the collecting duct to the blood capillaries. Then collecting duct to the blood capillaries. Collecting duct to the blood capillaries. The collecting duct to the blood capillaries. Next one, the mechanism of the concentration filtration. Now, what is the mechanism of concentration? See now, the mechanism of the concentrated filtration. What is mammals have the ability to produce concentrated urine. The mammals produce, human beings produce concentrated urine. The reason is more number of useful materials, sodium, the sodium and also the useful materials, nothing but glucose, amino acids, the small amount of proteins and also hormones, enzymes, vitamins, every materials reabsorbs into the renal tubule to the blood capillaries. But they releases into the more reabsorption means they releases to the concentrated urine. More reabsorption, that's the releases into the concentrated urine. But now in the concentrated urine, which parts are participated into the concentrated urine? Loop of Enels and vasa recta. What is the loop of Enel and vasa recta? Loop of Enel and vasa recta participated into the serum. I will show the next one. Two limbs of the English loop and opposite direction of the you can take English loop opposite direction. See the filtration in the opposite direction. The filtration is the opposite direction. Come to the counter current. Counter means opposite flowing between the it is the ascending limb and descending limb. And also counter current pattern, the flow of the blood, two limbs of the vasa recta, the surrounding of the loop of Enel consists of the what is this one? Vasa recta. In the vasa recta, the blood flows into the opposite direction. The blood is flows into the opposite direction, counter current, opposite flowing between the filtration is opposite direction. That is nothing but loop of English. It is nothing but vasa recta. In the vasa recta, opposite directions takes place. Opposite direction takes place between the opposite direction. Next one also connect the proximity between the proximity between the English loop and vasa recta as well as counter current helping the increasing the osmosis towards the interstitial medullary fluid. But basically, what is this one? See one, see here. The diagrammatic representation of the counter current mechanism. It is the opposite flow. It is the opposite flow. 300 to the it is filtration is the one is the downward direction, one is the upward direction, and also vasa recta. This vasa recta is surrounding of the loop of Enel, like this one. The vasa recta also surrounding of one is the downward direction, one is the here. The urine is filtration to the, for example, what is the concentration? 300 increases 400 to the 600 to the 800 to the 1000 to the 1200. They maintain the 1200. More reabsorption takes from the electrolytes. The again the concentration is decreases. Coming to the here 300, 400 blood. normal salt concentration in the blood 300, it increases 400, okay, 600. These sodium chlorides, they reaches into they enter into the blood now. They enter into the blood. That sodium concentration is increases. Whenever the sodium concentration increases, the water now, see here water is produced now. This water is enter into the here now. Whenever the water is enter means now, the water is reaches means now, the salt concentration is again decreases now. The salt concentration decreases again. In this side, the along with water, some urea also maintain. Urea and the sodium chloride to maintain the salt concentration. This is the counter current mechanism. Thank you so much.